I'll start. Today is the last day of my lecture, and so I forgot to make the slide for your assignment, and so maybe I will make the slide lunchtime, lunchtime, and then so I'll tell you. Okay, and today so today, at last, I'm going to talk about microfluidic devices for alternative method for animal testing or human testing, a little bit. Anyhow, so first, so I'll start. So by the way, can you find a frog on a small coin? Can you find? Yes? Yeah, so color is almost same, and it's a little bit difficult to find it. And here. So this is a real frog. You see? And this one. So it's a pencil. A very, very small frog. So this is the world's smallest amphibian. Very small. I want it, but I don't know where. Maybe some foreign country, so, but I don't know. But and then next. So it's a chameleon. This one is a world's smallest reptile. You see, it's also a very, very, very small. I want it too, but very small. It's very pretty. Yeah. This one is also I forgot. Amazing. So I forgot the name of the island or country, but, near, very close, close to Africa. But, I forgot the name. Very special island, not so small. And then next, can you, maybe you can imagine next slide. Next slide is, of course, world's smallest mammal. World's smallest mammal here, this one. It's pygmy jaguar, kind of. So there are several world's smallest mammal, but mostly, so these type of the rodents. So, length of the body about around 2 centimeters, but it has a very long tail. So, including tail, of course, not so small. But anyhow, if removing the tail, he can survive. So real body is just 2 centimeters, very, very small. I don't want it because maybe jumping, and it's very hard to maintain. But I want these livings, so chameleons and the frog. It's very interesting. And then, so we are going to make the world's smallest mammal culture system. This one. So maybe you can't imagine how small it is, but I'm going to make this system, and then so maybe firstly, so in the first maybe this system would be big, but and then afterwards I'm trying to become smaller. Firstly, but anyhow, so today's talk about like this, and so microfluidic device technology. So microfluidic device technology is based on the technology developed in semiconductor field. So now in Taiwan, so semiconductor field is very, very so active now. And that's why I'm coming here talking about that. So always I'm looking for the person working on the semiconductor company in Taiwan. But anyhow, so this microfluidic device technology is completely based on the technology developed in the semiconductor field. And combined with other processing techniques to make microelectromechanical systems. MEMS of course, MEMS comes from the for example, M of micro and E of electro, M again of mechanical, and S of systems. We call MEMS. And sometimes analytical systems are also introduced in this system. And then so sometimes we call a micro TAS, micro total analysis systems. So MEMS technology covers from the centimeter to nanometer order. So okay, centimeter or nanometer order, some here. Okay, and also yeah. Okay, and mostly we are making the microfluidic device made from the PDMS. So please remember this material, PDMS, polydimethyl siloxane. So yeah, you can take a photo, but maybe you have a handout, right? Handout, so material. Maybe, so I forgot to add these slides. Yeah, sorry. Yeah, it's okay. Yeah, okay. I forgot it. So PDMS is very convenient for making the microfluidic device because PDMS is a high gas permeability. It's very convenient for the supplying the oxygen. But of course, now I'm talking about the co-culture model of epithelial cells and intestinal bacteria. So intestinal bacteria, most of intestinal bacteria are anaerobic. So, but of course, epithelial cells need oxygen. So completely opposite properties. Then so we have to control the anaerobic and aerobic environments. But the PDMS is a high gas has a high gas permeability. Gas means of course oxygen and carbon dioxide and hydrogen and so on. and nitrogen also. And then so it's quite difficult to control anaerobic and aerobic environment by using the PDMS. It's a very big disadvantage. But most of person means a scientist or anyhow, so microfluidic device field persons, they are using, so they are making the microfluidic device from PDMS because it's a very convenient. And most of persons are not so interested in a co-culture model just now. And then, but anyhow, and also PDMS is external hydrophobic, and sometimes we have to treat with plasma for the oxidation to become hydrophilic. And also I forgot to add another one. Maybe yeah. And anyhow, so PDMS is a very convenient, and also this so using these properties of PDMS, this is an implantable medical device here, medical device used for the purpose of the birth control. So a small flexible tube containing the progesterone is a drug. It's inserted under the skin, usually here. And in the upper arm, the device can be easily, and the device can be easily removed when it is no longer needed. And then this is a very good example of using the PDMS. But so anyhow, and then so MEMS technologies such as gyro, sensor, and so on, and then can be set in the chip. And then so I don't know anything about gyro principles or anything like that. Anyhow, so other technology also can be installed in the microfluidic device. And of course, the microfluidic systems can have more several component technology, and for example, to different solution, so it can be so mixed. Can of course can be mixed and mixed by the turbulent flow, and or kept different flow in same line, I guess. Same line by laminar flow. And this picture is from Kitamori, and so he was retired from the University of Tokyo, but he is now Taiwan. Maybe he's now a professor in Taipei University, maybe, I forgot, or Tsinghua. Anyhow, so no spot. But I have never met in Taiwan, so I don't know. But he's now a professor in Taiwan now, after retirement of retirement from the University of Tokyo. And okay. And then also there are mainly some advantages of MEMS, such as miniaturization, so to make very smaller, and also batch process without framework, and functional integration, the gain of novel functions, such as this one. So concept, this one is a concept of microtask, or anyhow, so concept of a microfluidic device system. And width length is a few centimeters or less. And many technologies such as a micropump and magnet valve for controlling flow rate and the mixture, and also heating system provides a chemical reaction and some measurement on the chip. So this very small chip is really same as a big laboratory. So sometimes we call lab on a chip. So only one chip contains all of instrument in the laboratory. And so it's amazing. And then so 30 years ago, I heard and I read these papers written by Kitamori, and Dr. Kitamori, and oh, good. I wanted to make this system because I have many cell culture systems, and also we have many techniques for manipulating the human iPS and the human ES cells and so on. And then so at that time, Dr. Kitamori, he is a chemist, and then so just he introduced the chemical reaction laboratory, and then so but now so I wanted to put the cell culture system here and there, and then but in recent, this is a so Newsweek journal. Newsweek journal introduced body on a chip. So body on a chip, and you can see body systems such as lung, stomach, and the pancreas, and intestine, and so on in the chip, and so on the chip, and so this chip is pinched by pincets. So you can imagine so pincet is like this. So this pincet so is of course small, and then very, I think very good picture, awakening imagination of a very small body system on a chip like this. Of course, we had already started to make the device, and anyhow, and also in recent microfluidic device technology has been developed, and then so we are anyhow so trying to culture several cell lines on the device, and for example, so here, so I can't read it. Maybe here they describe liver, and here I can't read it. Maybe some organs anyhow, and here I can read marrow or something. Marrow, it's a very strange. But anyhow, so they just culture on the culture and the cell lines, each cell lines on the chip. So, but they said so living system on the chip. So, but they use just a cell line, derived from the tumors or from sometimes the primary cells. But no adequate cellular polarities. And then so those cells have absolutely weak physiological functions, maybe, or no function compared to real one, of course. And then so maybe just cell lines are culture on the chip. So I don't agree with their paper. They describing the living system on a chip. I think not so good. But now also the day before yesterday and yesterday, I explained we are making the tissue structure from the human iPS cell or mouse ES cells. And these are very functional. And so I wanted to these organ tissue like culture system on the chip. Anyhow, so there are other papers also. They said they succeeded to culture several organ culture on the chip device, and but just using the small intestine was epi-intestinal TM. So maybe they bought these cell lines from the company. But I think it's primary. I forgot, but I read maybe it's a primary cell. And the liver hepatology is a tumor cell line, hepatoma cell line, and the primary sterate cell mixture, just as sterate cell. But I think to construct the hepatic tissue, not only hepatocyte and sterate cell, and also endothelial cell, especially liver sinusoidal endothelial cells, necessary. But they didn't add sinusoidal endothelial cells. And they cultured skin, juvenile cells, and kidney. Yeah, but this is not function. And then so first our one of I will introduce several organ on the chip. And then first one is the heart on the chip. As I told you yesterday or the day before yesterday, and then so we could differentiate mouse embryonic stem cell to cardiomyocytes spontaneously contracting. Then so through our embryonic body like this, and then so in this paper, so we implanted these mouse embryonic stem cell derived cardiomyocyte into the mouse, and then we succeed. And then anyhow, so it's spontaneously, automatically, so contracting and addition of isoproterenol and feeding high, and then block the protein by protein and like this. So using the device, and then so you can we could get electrocardiogram like this, and it's very convenient. But now, so this one just we bought this device. So it's not so special, but of course to get electrocardiograms like this, very special software is necessary, and so it's quite difficult to make by ourselves, and so just this one. And but the next one, so liver. So liver, you know, so liver plays a crucial role in the metabolism, glycogen stock, protein synthesis, and detoxification for survival. So it's a very important and necessary roles. And then most important organs. And also, so as I told you, maybe just before yesterday maybe, and so it is very difficult to maintain physiological function. I mean, in primary culture. So urea synthesis is one of the representative hepatic functions. So I told you already, hepatocyte has multiple, really super multiple functions. Not only the synthesis of synthesized urea, also I told you also metabolism and glycogen stock and detoxification and producing other proteins such as albumin and coagulation factor or something. And then so, but so this is one of them, and but so this function is anyhow so dramatically decreased day by day. So it's really difficult to maintain the hepatic functions. So other functions also. Maybe so if I try to explain to show you the other hepatic functions, so I need 1 hour, and so just so I present one of them, urea synthesis. And then so I told you, so this is a hepatic logo histological cartoon. Then so here portal vein connected to intestine. Then so nutrients and drugs and toxins also comes from the intestine to portal vein. And then this flow direct to the central vein, and molecules incorporated into the hepatocyte, and the hepatocyte monitor, which is a good one, or this one is a bad or something. Then so detoxify or modify and so on. And then so it's very important. And so this we call sinusoid. So sinusoid consists of sinusoidal endothelial cells, and these are hepatocytes. Okay. And this is a real histology, and this is a central vein. No, this one is, I think, portal vein. Just not so good picture. And anyhow, so hepatocyte has 3 cellular polarities. Here basal face, and here lateral face, and here this area is the apical face. So apical face always to outside, and basal face faces to inside of the body. Inside means in so yeah, mainly to face to blood vessels. And the green one is the endothelial cells, and here also this one is a hepatocyte. And then so yesterday I explained so we make the hepatic tissue from the mouse ES cells by using the embryological also liver organogenesis information like this, and then cardiac mesoderm firstly appeared from the outgrowth of embryo body from the embryo stem cells, and then cardiac mesoderm produces some cytokines to initiate to the hepatogenesis, and by receiving these signals, so ventral foregut endoderm migrated into the septum transversum, and then to form the liver buds. Liver buds consist of hepatoblast and endothelial progenitor cells. Exactly maybe sort of sinusoidal endothelial progenitor cells. Okay, then these liver buds are expanding. Expanding means proliferating, growing, and then finally the liver is completed. Okay, then so firstly we prepared embryo body consisting of 3 germ layers such as endothelial, endoderm, mesoderm, and ectoderm, and from the mouse ES cells. And then so these embryo body, around 50 embryo bodies culture altogether, and then the outgrowth of this is the outgrowth of embryo body. Then so part of the outgrowth of embryo body, it's a mesoderm, and then so mesoderm developed to the cardiomyocytes, and then cardiomyocytes producing the several cytokines. Then to initiate to the so signaling send the signal to the endodermal area, and then so endodermal area receive these signal, and then making the so developing to the hepatic tissue like this. Then so this is the experiment, and then this one is our mesodermal cardiomyocyte, and automatically beating like this, and then around neighbor these area. So here albumin positive cells appeared, and then to form the cluster. And then so it means proliferating, and then so albumin positive cells is might be hepatoblast, and then so these hepatoblasts are proliferating. So hepatoblast, it's a stem cell or progenitor cell. It's a little bit unclear status, but anyhow. And then so interestingly, we detected hepatic morphogenesis in the embryo body outgrowth, and consists of red ones are hepatocytes, and green ones are CD31 positive cells, it's endothelial cells. And then so you can see the endothelial cells were seen to be migrating into the colony of albumin positive. So hepatoblastic area, and it's like a very similar to liver buds in the liver organogenesis. And then finally, these cells, both cells, hepatoblast and hepatocytes and endocellular cells are proliferating and expanding like this. This is the 23rd stage after the embryonic body formation, like this. And then so yesterday I showed many functions in this mouse embryonic stem cell derived hepatic tissue. And then but today I'm going to introduce these hepatocyte, these hepatic tissue tissue was cultured on a dish, on a device, so microfluidic device on a chip like this. So to culture the so constructed liver tissue in a microfluidic device, care must be taken with the medium flowing into the tissue. So the medium needs to flow in gently, not like this, gently, so that it flows in broadly like Niagara Falls, something, and exists. And also exists. It's also from the entire ceiling here. Ceiling here. You use the entire ceiling. Okay. Then so we invented this microfluidic device, and this slide shows a detailed description of this device. Maybe you don't need this information, just look at this one. Anyhow, so we have to make finely like this. Yeah, but I gave the, how can I say, idea and also the scale or something to Shimazu Corporation. And then so Shimazu Corporation made this device. Yeah, so just so I a little bit more thin or something, and also this slope is necessary, and please make the slope and something. And I want to put the mesh on the ceiling to avoid the debris. Debris cannot go to the channel, because the channel is very small, and so if the debris goes to the channel, and then channel is maybe stuck or something, then it's so no more continue. And then so just so I order, and then so Shimazu Corporation always trying to make this system, and then finally Shimazu Corporation and our group succeeded to make this device. Then, so this slide shows hepatic tissue on a chip like this. So previous one, so we differentiated differentiated mouse embryonic stem cell to the hepatic tissue on the dish, but this one is we succeeded to make the hepatic tissue on a chip of that device. So anyhow, so you can see the endocellular network and the hepatocyte like this. And total liver chip system is like this. So chip is very small, but the total system is very big, bigger than mouse. bigger than a mouse. So, but if we maintain 100 mice, we have to provide around 20 kgs. So we need more space. On the other hand, in the case of our device system, so it would be easier to make a high throughput device on the same area here. Maybe so 100 is okay, so 10, so 1,000 is okay. Maybe so not so difficult. For me, Shima's corporation, they have to be harder to work to produce these devices. 1,000 devices. Oh no, maybe he complained maybe to me, but anyhow, so something like that. So this is the device, and here at that time, so syringe pump supplied medium, but now we are using the micro perista pump, a very small one, and then so we don't use anymore. Use it anymore. So and then so gradually becoming a smaller system now, and here dissolved oxygen concentration meter here, and the pH meter, and also so we can measure the pH change and or dissolved oxygen concentration change also here, not here, so here, so sublime from this chip. And also we measure the first urea synthesis. And so urea synthesis is a hepatic function, so as I told you just now, and so anyhow, so other liver functions are almost same. So consider urea synthesis capacity as a liver function. Now, so by incubating with the device and flowing the medium, the liver function increase very, very much like this compared to the non-flow. So this one is also, so this hepatic tissue also cultured on the device, but no flow, no medium flow. But by addition of the flow, so I didn't mention the speed of the flow, but I forgot maybe 40 microliter per hour or something. It's almost a similar flow rate in the sinusoid. Almost. Okay. And anyhow, so and then next, we are committed to the idea of liver tissue, and just now I introduced second one already, and but there is another approach also, and therefore I'm going to explain the second one. First one. Talk about the first one. This one. So firstly we established the GFP expressing immortalized endocellular cells. So it's endocellular cells, but so honestly speaking, it's a little bit different from the liver sinusoidal endocellular cells. So these cells unfortunately have no pore, so not fenestrated. Okay, so liver sinusoidal endocellular. Do you remember yesterday's talk? So sinusoidal endocellular cells have holes, many holes. We call fenestrated, and to intake the nutrients or toxin and so on. But this one is not. Just these endocellular cells might support to make the cellular polarities in hepatocyte. Then anyhow, we firstly established the GFP expressing immortalized endocellular cell lines. We call GH7, and so GH7 was tried to be cultured on several materials, such as collagen coated, fibronectin coated, or laminin coated, poly-D lysine coated, and or EHS gel coated. So I call it EHS gel, but generally EHS gel is the same as matrigel. Okay, matrigel. Then so, and the EHS gel is the only one of these ECMs in which the endocellular cells from such a network structure here, like this. Okay? Like this. To form the network structure. It's a very special. And also, maybe so within 4 or 5 hours, these endocellular cells start to form the network structure. And part of the, here, for example, network structure from the tube, like this. Okay? Tube. Not tube. So here, these are tubes. Okay? Like a vessel. And then red fluorescent primary hepatocyte from the mouse, from the red DS red transgenic mouse liver. We prepared like this, and then so red fluorescent primary hepatocytes were seeded on this GH7 network structure. Okay, and here like this, it's just a start time like this, and then these hepatocytes migrating toward the endocellular cell network structure like this. We have already modified the structure. Now, of course, we added sterate cell also, and also we modified a little bit matrigel also changed a little bit, and anyhow, so it's this one we call hepatic sinusoid-like structure tissue, and also using a primary hepatocyte and the endocellular cell on the EHS gel like this. And also we tried to make the human sinusoidal structure by using the human iPS cell derived endocellular cells like this. So we call IVL, human iPS cell, like this here. So, and then we are discussing. We at that time we discussed how to use this human iPS cell sinusoidal culture model. And so, firstly, I wanted to infect. I wanted to make the HBV infection culture model because at that time we didn't know the HBV receptor on the hepatocytes at that time. But my hypothesis was this HBV receptor should express on the special phase, I mean maybe a basal phase of the hepatocyte. And then so my hypothesis was agreed from the government, and then so I got at that time big grant, and then so started. And then during this project, then Chinese group found the HBV receptor, this one, NTCP. NTCP is a sodium taurocholate for transporting polypeptide. It's one of the transporter expressing on the basal phase of the hepatocyte in the liver. Then so they discovered this HBV receptor. Then so my hypothesis was also right, and then I could continue this project. Then you see not only the NTCP transporter, also other transporter also. These locates specifically on the basal phase, and for example MRP2 and BSEP, these transporter expressed on the apical phase. So that's why cellular polarity must be very, very important. Then so when we try to culture the cells to mimic the organ tissue, then so we have to consider the cellular polarities. Okay, and this one. Then so here red line. Look at the red lines. This is our IVL derived from the human iPS cells. And then so NTCP expression. NTCP is a HBV receptor. And then so HBV receptor expression keep the long period like this, compared to the just a human iPS derived hepatocyte, and HUH7 is a hepatome cell line. So no expression in the HUH7, and but iPS cell derived hepatocytes have you can find the expression of NTCP, but compared to our device, our tissue system, tissue culture system, then this one is very low, like this. Then so now we are ready to infect the HBV. Then so we challenge the HBV to our device, our tissue culture model, and then so the red one. So we could detect the full genome RNA in the cells, and then so increasing, and also correct the media, and then correct the media, and then so correct. It means that to correct HBV particles in the medium, and then also high. So we succeeded to make the HBV infection system on the human iPS cell-derived hepatic tissue, not hepatocyte, so hepatic tissue, like this. So then, but then so we try to make the microfluidic device system for HBV infection system like this. This one. Firstly, like this, and then so circulating again. Here, this one. So liver chip can be applied for a virus infection, but it had to be robustly fabricated because if the medium leaked, for example here and here, leaked, so it's a very dangerous because HBV contamination occurred, and then so we have to make robustly, okay, and then so but it's a little bit yeah, okay, and then so anyhow so focus on the hepatic tissue structure. It's almost same, so just change. Oh, no, just and then also I'm going to introduce another liver tissue chip. Anyhow, so this is the histology of the liver tissue, and then you see it's a little bit complicated and a little bit too small figures. And here this one, so I wanted to introduce firstly intestine device, but on the other hand, so I want to talk about intestine device last one, and then so, but so this system by using the intestine microfluidic device, and so we made a hepatic tissue device also. Okay, and then so you see this is a porous membrane here, and here upper channel, and here lower channel. So channel is a medium flow, okay, upper flow and lower flow like this, and also here porous membrane. Then so endothelial cells cultured on the porous membrane here. And then also RH4 is a hepatocyte. So hepatocytes cultured under the porous membrane and also on the bottom of the device here. So here is bile canaliculi like this. Bile canaliculi. Maybe back here. So this one. So bile canaliculi. So between the hepatocyte and the hepatocyte. Okay. And here is the sinusoid. Sinusoid, of course, of endothelial cells here, and then so here. So this is a bile canaliculi, and here is just so face to the endothelial cells and so sinusoid. So we try to reconstruct sinusoid and the bile canaliculi. So flow is reverse, okay, like this. So opposite direction. And this one, device. So electrodes here, but later I will explain. So here, so red one hepatocyte, and the green one the endothelial cells. Like this. So rolling like this. So this is the bile canaliculi like this, okay. And here also I show you like this, okay. And then so this hepatic liver device should have a cellular polarity, 3 cellular polarities in the hepatocytes. And so we check. So this one. So this is the hepatocyte. You see, this is the hepatocyte. Then here basal phase. Okay, basal phase. Because neighbor endothelial cells. Then so based on the basal phase, here OATP1B1, it's one of the transporter. Then this transporter locates at the basal phase. Okay, and here CDF, just not so good picture here, just. Yeah. Okay. And then so here is the ABCC2 transporter. So this ABCC2 transporter locates at the apical phase. So lateral phase just adhere to the other neighbor hepatocytes. Okay. Then so if this liver tissue device would have 3 cellular polarities. So CDFDA is one of the molecules. So CDFDA has no fluorescence. And the CDFDA is incorporated via their OATP1B1 into the cytoplasm of the hepatocyte. Then, so esterase is an enzyme. So esterase exists in the cytoplasm of a hepatocyte. And then, so this esterase digests CDFDA, diacetate, so acetate, this ester bonding digested. Then so become to a CDF. So CDF has green fluorescent. Okay, CDF fluorescent. And then so CDF can be excreted via ABC2 to the outside here. Okay? Then this one. Oh, this one is also not. Just a moment. So CDF okay. So CDFDA was added from the upper channel, and then so CDFDA incorporated into the hepatocyte here, and then CDFDA was would be digested, and then to CDF, and the CDF it would be excreted into the outside here, the lower channel like this. Then so you see, so this is a basal medium, so basal medium from the basal. Then so of course no reaction because CDFDA has no color, so no fluorescence like this here. So one after post 1 hour, 2 hours, 3 hours, and then so on the other hand, apical. Apical means here, lower channel medium. So you see, 2 hours, 3 hours, you can see the green fluorescence. It means like this. So CDFDA incorporated into this hepatocyte and then excreted to the outside, to the lower channel, like this. So using this device, so we can measure these dynamics, drug dynamics. So it's really convenient and for the alternative method of animal testing or human being testing right now. Yeah. So microfluidic device, it's very convenient, but anyhow, so there is a very big disadvantage because operator means so including you. So user needs very special skill to operate this device. But our device is not so complex, and then so maybe if you have so after you receive the training for 1 or 2 months, and then so you maybe you can operate this device. Indeed, this data this data was produced from the undergraduate student. She succeeded to get this data. So she received just maybe 1 or 2 months. Then finally she can operate this device. So, but anyhow, so now so we have only so just one system, and so always students waiting for their turn. So, but anyhow, so okay, so then yeah, and then so next I'm going to talk about the intestine device. Then so first I have a so maybe so I'm going to receive the question first from you. Do you have any questions? Do you have any questions before moving on intestine device? Because it's a basic one, and so so could you follow my talk? If you know, so please ask me now. No question? Did you understand? Oh, okay.
One question. Yes. I see in the handout there is the picture for the device, like endothelial cell is on the up there, and there's the cells
sorry. So
okay.
which slide?
Mm, here.
Oh, this one.
Yeah.
Just a moment. So this one.
Yeah.
Yeah.
And there's the median for these two kind of cells is separated, and it's why is it separated? Yeah.
Ah.
So this is so mimicking the hepatic tissue structure. And so here, maybe. So here, this one corresponding to the sinusoid, and here corresponding to bile canaliculi. So I show you the this figure is a good one or not. Yeah, I tried this one. So here sinusoid. So sinusoid from the portal vein to the central vein, right? Okay. And then so this is the sinusoid. So sinusoid consists of endothelial cells. Endothelial cells. So now we mimic here. Yeah, here is best, I think. Here. You see, so endothelial cells, and this is a sinusoid flow here. So the direction is opposite, but anyhow, so here, and this one. So she drew the bile duct, but anyhow, so bile canalicula here. Okay, so bile canaliculi locates between the hepatocyte and the hepatocyte. Okay, and then we so here this flow corresponds to sinusoid, because here there are many so there are endothelial cells, and then so here hepatocyte layer, and then between the hepatocyte and the hepatocyte, so I told you that so bile canaliculi. Yeah, so that's why they're separated. So it's okay?
Yeah.
Okay. Yeah, it's a very important point. So do you have other questions? Other questions? No, it's okay. Okay. Yeah, yeah. Right.
And in the slides shows the
Mm-hmm.
the flow rate of this glycol fluid, and I wonder how to define the flow rate of it.
So flow rates problem is very important, and then so in this case, so 40 microliter per hour, we decided. Of course, we tried other speed flow rate, of course, 100 micrometer 100 microliter per hour and 200 microliter per hour. But so higher than 40 microliter per hour, it's possible by using this syringe pump. But lower than 40 microliter per hour, it's impossible by this syringe pump. But fortunately, we are very happy. So we calculated the speed of the sinusoidal flow in the liver around 40 microliter per hour. Oh, lucky. Exactly speaking, a little bit different maybe, but almost same. And so I was very lucky, and so 40 microliter, so very good. And also just so we collect. So sometimes we had to collect the media from the device, and then so 24 hours, just maybe 1 milliliter or 1.5 milliliter, and so it's a very useful. Yeah. And so next, so I'm going to introduce the co-culture model of epithelial cell and intestinal bacteria, and so flow rate is different. Flow rates are different, and then little bit complicated, but also when I present these content at the international meeting, and always someone asking, why, how did you decide the speed of the flow? Always, so I received the question always. Yeah, but now it's okay because we have already published this so data, and then so now I put I could put real speed of the flow. But sometimes I can't tell them the speed because flow rates are very important. Yeah. Okay, and any questions? No questions? I hope you understood my talk. I hope it's okay. No questions? Oh, okay. So if you have more questions, please let me know. It's okay. But now, so let's have a break. So, okay, let's move on in testing here. So, just here. So I have already explained liver chip and heart chip and so on. But now I'm going to talk about intestine chip. So intestine has lots of bacteria, intestinal bacteria. So I think I thought co-culture of intestinal cells and intestinal bacteria should be necessary. Then because this is so maybe you know already. So here stomach, and is a small intestine, very long, and so maybe stomach. In the stomach, oxygen exist, but concentration of oxygen dramatically reduced along in small intestine because some aerobic bacteria resume the oxygen. Then finally, maybe here, maybe large intestine, so no oxygen, and then these bacteria, of course, are anaerobic bacteria. Okay, and anyhow, so we eat some food and containing three representative large molecules, such as carbohydrate, proteins, and lipids. And just one. Carbohydrate is polysaccharides, and carbohydrates are digested by the salivary juice, and to mono or bisaccharides in mouth, and proteins delivered to the stomach, and then proteins are digested to amino acids or small peptides by gastric juice. And finally, lipids, and finally lipid to glycerol and long-chain fatty acids by pancreatic acids. And then these monosaccharides or bisaccharides and amino acids and small peptides and long fatty acids and so on incorporated into the incorporated at the small intestine, then delivered to the liver, and then after liver circulating or excreted or something. So that's a very basic knowledge. Maybe you have learned at elementary school or something. But you know, many bacteria live in biosis in the human body, and for example, 40 trillion or more bacteria in digestive tract. The bacteria also, but our body consists of 30 trillion mammalian cells. So comparing the number of cells, mammalian cells, so it is no exaggeration to say that humans are composed of bacteria. Okay, and so do you, maybe I have so you have already answered, but do you know this monkey? I have a hobby. One of the many hobby, but so one of them is a journey. And so at that time, so I went to the jungle near Kota Kinabalu. So Kota Kinabalu is in Kalimantan Island. Kalimantan Island, you know maybe. And Borneo Island. And so I went to the jungle, and then to find these monkeys. So these monkeys, especially male has a very long, big nose. You see, the long nose, and a very big belly. So in this talk, don't look at big nose. Look at big belly Okay, and this one proboscis monkey in English. But we call this monkey Tengu-zaru. The sar Tengu-zaru. Sar is a monkey, and Tengu is how can I say? One of the devil in Japan. So he's living in the mountain, and they are having a very long nose, and so we call Tengu-zaru. And it's a very special face, and so I'm interested in this monkey, and then so I wanted to meet him, and then so finally I went to the jungle in the Kalimantan Island. And then anyhow, so this monkey is very special. So other monkeys cannot eat mangrove leaves. So these trees are mangroves. Okay, mangroves. And then so it's not so difficult to find these proboscis monkeys because just to look for the mangrove trees. So my guide told me. But there are many mangroves in the jungle. Most of the trees are mangroves. So, and so finally, we found proboscis monkeys. But anyhow, so other monkeys cannot eat mangrove leaves. But the proboscis monkey can eat them. So proboscis monkeys succeeded to escape survival food competition with other monkeys by eating the mangroves. Okay, and so they don't have to quarrel, fight. So they don't have to fight to death. Just eat mangrove leaves, and then so anyhow, so look at his belly. So he has a very he has a very long intestine to detoxify mangroves poison. So because mangrove leaves contains much toxin. And that's why other monkey, including human being also, cannot eat mangrove leaves. Of course, very bad taste, but anyhow, so contains a much toxin, and that's why the other monkey cannot eat. But very strange, proboscis monkey can eat mangrove leaves. So mainly, they eat mangrove leaves. And so because they have a very special intestinal bacteria to detoxify the mangrove leaves toxins. And here I visit Melbourne in Australia, and so it was not so difficult to find a wild koala. You see, just of course the city of the Melbourne, no koala. Maybe so you have to go to the zoo. So you can meet them. But anyhow, so suburb of the Melbourne. Then so also it's not so difficult to find koala, wild koala also. So I think. And then I found a wild koala, this one. But unfortunately, he or she, I don't know, but this koala living in the very high tree, and so resolution is not so good. But anyhow, so I found the koala. And so koala also escape food competition by eating the eucalyptus. I don't know that. So eucalyptus also I heard koala can eat specific sort of eucalyptus. So there are many kinds of eucalyptus in the Australia, and then so they can eat only a few types of eucalyptus. Then so I'm not sure. So anyhow, so proboscis monkey monkey has a special intestinal bacteria, but maybe koala also. So eucalyptus also contains much toxins, and so maybe koala has also. I'm not sure, but maybe a special intestinal bacteria to detoxify. But koala has also a special functional liver. So maybe it's detoxified and eucalyptus poison in the liver. But so I don't know. And here I visited Inner Mongolia. So do you have ever been in Mongolia? Mongolia grass field, very wide. So nobody over there, and no building, only grass, and also sometimes horses are running. Of course, with the human, but anyhow, so at that time, so maybe it was 4 a.m. or something, very early morning, because I stayed in the in a very special Mongolian tent, and so unfortunately, I couldn't get hot water in this jar, and then so I couldn't sleep well, and then so I woke up at early morning, 4 a.m. and then so walked around the grass field, and then so horses, of course, already so wait, and then so eating the grass, and also you see. He eats not only the grass, also you see what do you know what is this? So maybe so pieces. So horse, maybe one or other horse, maybe other horse one, maybe pieces, and then so he or she, I don't know, but anyhow, so this horse eats someone's pieces. But I think compared to the other pieces, it's like a very good one. Maybe I could imagine it's maybe tasty or something, but I don't know. But anyhow, so we call this coprophagy. So coprophagy we can observe in the other animals also, mouse, and of course koala also. When koala is baby, infant, and then so they eat feces from the mother. Then to get special intestinal bacteria, I think. So coprophagy is very important. Of course, we maybe I want to believe I have never eaten pieces, but anyhow, so but it's very important for the wild animals. And then let's look at the mouse. So this one shows the digestive tract tract of a normal mouse. So so SPF. SPF is a specific pathogen-free mouse. It's this mouse breed under the normal condition. So you see. So of course conventional mouse also, but anyways, this one is a specific pathogen-free, so not germ-free mouse. So they have lots of bacteria inside of the intestine and also the on the skin also. Okay. Anyhow, so SP mouse breeding under the normal environment. On the other hand, germ-free mouse. Maybe I sent already your handout, right?
Yeah.
Yeah. So maybe so you have already answered. So in germ-free mouse, so I wanted to ask you, how do you imagine the digestive tract of the germ-free mouse? I was going to ask you, but you already have the answer on the handout. So show it quickly. And this one. So in germ-free mouse, you see a very abundant pieces. Okay, very, very abundant pieces. And food was, you see this color, it's, this color is as the food, mouse food color, mouse food color. And then so food was not fully digested, okay? So because there was no intestinal bacteria in the intestine, of course, a germ-free mouse. And so intestinal bacteria, so it means intestinal bacteria play an important role in the digestion. Therefore, co-culture of intestinal bacteria and intestinal epithelial cells are necessary. That's why we are trying to establish the co-culture model of epithelial cell and intestinal bacteria. Okay, and maybe you need more information. And then so not only the food we eat, but also medicines, especially for herbal medicine, to be effective and absorbable. So their digestion by intestinal bacteria is important. So therefore, co-culture of intestinal bacteria and intestinal epithelial are necessary. I hope you can understand. So, for example, so this is the herbal medicine. So herbal medicine contains a very complex components. You see. So, of course, so herbal medicines are very popular in Japan and of course in China and in Taiwan maybe. So do you know herbal medicine? Okay. Herbal medicine. Okay. Yes. Kampo. We call kampoyak. So Chinese medicine. Okay. Chinese medicine. So herbal medicine. So I don't know so I'm sure in Taiwan. So I can call these drugs as Chinese medicine or Taiwanese medicine, or I don't know. And that's why so I described herbal medicine. I think I hope this expression should be fair. I don't know, but anyhow, so herbal medicine, and but complex components, and mainly glycosides form. So glycosides are very hardly absorbable, and but we take the herbal medicine as glycosides. Then, but these herbal medicines are effective because intestinal bacteria, they have glycosylation enzymes, and then glycosides, they receive the glycosylation to effective molecules, and also absorbable. So there, I gave one example, licorice in English, but maybe you see, I don't know the pronunciation. We call kanzo. So your pronunciation is, what do you say?
Pretty similar.
Hmm?
Pretty similar.
Similar.
Yeah.
So kanzo, correct?
甘草.
Tansao.
Yeah.
Tansao. Okay. Tansao. So I asked Professor Wang to show the root of cancer. And then but he doesn't have, and then now he's asking his friend, a medical doctor, and I hope next week I will see cancer. Maybe. But anyhow, licorice, and licorice. So of course, licorice is the king of herbal medicines. So there are many herbal medicines that contain the licorice. You see, I don't know the pronunciation, and so maybe you can recognize these herbal medicine. I prefer the herbal medicine rather than Western medicine. Always, sometimes I took these herbal medicines. And also, 3 weeks ago, I was in Hamburg, Germany. And then of course, for international meeting. And so I found licorice infused chocolate like this in the supermarkets. So licorice is licorice. So licorice is not German. So it's a Swedish spelling, licorice. Because this, so I found this sweet in the supermarket in Hamburg, but this chocolate made in Sweden. And anyhow, so North Europe, very severe winter, and so maybe they are afraid of the catching cold. And then so always they are taking these type of sweets containing the liquids. And I have been in Finland, and then Finland also very popular. So most of sweets contain this licorice. Very strange. Most of them. Of course, chips, potato chips, and American snacks. So and snacks do not contain the licorice, but local sweets, most of local sweets contain the licorice. Very strange. And also, I found very famous sweet, not sweet, but anyhow, so just, how can I say, the local sweets, they say, in the Finland. I forgot the name of this, but it's a very, contains a pure licorice component. There's, I tried to eat it, very bad taste and very bad smell and very strange. Yeah. But anyhow, so at that time, so I didn't take a photo of it. So I bought this chocolate because at that time, so I brought my PhD student for attending this international meeting. Then, but unfortunately, she suffered from the coronavirus infection. And the first day appeared, and then so I'm okay for something, and then but I forgot to bring some drugs, and then so I went to the supermarket and then looking for something, and then I found licorice, and then I hope this one it should be effective. Then so I took every day. Maybe it's okay, I think. But anyhow, so licorice. And then so back to the licorice talk, and then so licorice, of course, contains complex components, but one of the components is glycerylretin. So it's a major component of licorice. But this one, of course, glycoside or something, and then so we have already known glycoretin. It's non-effective and very hard absorbed. Then, but most of intestinal bacteria produce beta-glucuronidase. So beta-glucuronidase digest the glycerylretin to glyceroretinic acid. So glyceroretinic acid is effective. And so effective means increased increased intestinal motility and also anti-inflammatory, anti-inflammation, and then so it's very effective. So so intestinal bacteria. So herbal medicine also completely depends on the intestinal bacterial activity. So for the drug screening, we have to consider the intestinal bacterial activity. So don't neglect the intestinal bacterial activity. So it's very important. So study the metabolism and the dynamics of drugs by co-culturing intestinal bacteria and intracellular cells. However, when the mammalian cells are cultured on a dish, we have to avoid bacterial contamination. Once some bacteria have fallen into the culture of the medium of the culture, so bacteria grow much faster than the mammalian cells. The bacteria produce a lot of lactic acid and also toxins and consume the nutrients. Nutrients are very important for the mammalian cells also, but bacteria growing fast, and so they are consuming the nutrients, and anyhow, so very bad situation for mammalian cells. So how to do it? For the co-culture of the exterior cells and bacteria. And then, so if the number of bacteria can be controlled, and also toxin and waste can be removed by such flow, medium flow, so such a fresh medium flow, it will be possible to co-culture mammalian cells and bacteria. So in order to do that, so microfluidic device technology has a very big advantage. So fluidic device, okay? And then, so anyhow, so yeah. So here, and then so if we could control the number of bacteria and excrete toxins and waste through such a flow, and so I thought we might be able to co-culture mammalian cells and bacteria on the microfluidic device. And this is the typical schema of our microfluidic device, our device here. And you can see here porous membrane, and on the porous membrane there are so exterior cells, okay, exterior cell layers formation, and some cells produce the mucin. So here the mucin layer. So mucin layer is a wall, is a barrier against bacteria invasion. So this mucin also very important. And you can see mucin layer, and also you can see the microvillus here. So here, apical face, okay, apical face of exterior cell layer, and here, basal face of exterior cell layer. So you can observe the microvillus on the apical face side, apical face side. Okay, so these are very important microvillus, and also mucin layer here, and also exterior cell layer should form the tight junction here, tight junction. Okay, tight junction. Tight junction is also very important for the avoiding the invasion, avoiding the bacterial invasion into the body. So completely, so this one is a barrier, complete barrier. Okay. So anyhow, so I will explain again. This is our device. And so this microfluidic device has a, anyhow, so porous membrane, okay? Porous membrane is having the many holes, okay? Holes. And so, two medium flow here. Sometimes on my several slides, flow direction, or sometimes the opposite, but then here from the right to left, okay, flow channel. And so epithelial cells, of course, are cultured on the porous membrane. The cell culture area can also be observed under the microscope here. So because this device is completely transparent, and then so we can observe the cell culture condition under the microscope. Yeah, of course, from the naked eye, but this one is very small. Okay, and so also there are two electrodes are attached to the both channels. Both channel is upper channel and the lower channel to measure the transepithelial electrical resistance level between the upper channel and the lower channel. It means in order to monitor the tight junction of epithelial cell layer. Okay, so we call transepithelial electrical resistance level. We call TER, T-E-E-R, T-E-E-R, TER. So the TER correlates with the level of a tight junction of our epithelial layer. So if the TER level is high, so tight junction should be strong. But if the TER levels decrease, tight junction may be broken. Okay. So cell observation and so on. And this one is a simple system. So here the pump. This at this time, so this pump is still a little bit bigger, but next, so it should will be smaller. And here inlet medium, so inlet flow like this, and supply the medium. Supply the medium by this pump here, and this one I don't know which one is upper channel, but maybe for example upper channel, and then outlet here. So we can collect the medium also, both from the both channels, and here connected because can measure the TER level. So, cell source of intestinal epithelial cells for intestine shape. First one is the cell lines, CACO2 and C2BB1 cell lines. So, and human primary enterocytes also available, and also human iPS cell derived intestinal epithelial cells. Example, FI-seq. This is we can buy these cells, human iPS cell derived epithelial cells from the Fujifilm company. So Fujifilm was sold. Fujifilm company sold had sold film for the camera. Maybe so you don't know the film. Maybe so already so you have just this digital camera. But when I was for example student, so at that time we didn't have this digital camera, and so we used film camera. And then so film camera is very special, and then at that time Fujifilm and I forgot Konishiroku and also Kodak. These are at that time these are three major film companies. And then so Kodak now disappear, I don't know, and Ponishirok also disappeared, and Fujifilm succeed to survive because they stopped selling the film. They changed to the medical field, and then now they are selling these exterior cells. Human, they succeeded to establish the differentiation system from the human iPS cells to intestinal exterior cells in the large scale. And then so now we can buy these human iPS cell derived intestinal exterior cells from this company. Of course, now maybe you can buy these type of exterior cells from the other company also. I don't know the Chinese company, so Acro Biosystems also is selling these type of cells. But I don't know my previous yeah. And also human iPS cells derived intestinal organs. So yesterday I introduced the organoids, our intestinal organoids. So intestinal organoids, and then so it's almost very similar histology to our real intestine 1, but it's not useful for the co-culture system of intestine organoid and intestinal bacteria, because inside of the core organoid is apical, and then so we have to inject. Of course, we have to inject intestinal bacteria inside of the intestinal organoid, but of course intestinal bacteria growing very fast, and then so maybe these bacteria killed organ, and then so it's impossible to co-culture. But I'm interested in intestinal organoids because as a cell source for this device. Then so we got intestinal organoid, and then dissociated to the single cells, and then so these single cells will be seeded on the porous membrane. Then we wait for the compression of the tight junction on the exterior layer. Then so we can use it. So now these cells are available, and then sometimes we are using these cells, but still very expensive. And then now I almost so I have used the C2BB-E1 cell lines, because the pharmaceutical company also use CACO2 or C2BB-E1 cells for the investigating the drug dynamics, so absorption and excretion. Then also metabolism also, and then so we are now looking at the pharmaceutical company for the application. Then so I decided to use C2BB-E1 cells. Oh yeah, C2BB-E1 cells. So C2BB-E1 cells were seeded on the from the upper channel, and then adhere to the porous membrane to form the tight junction, like this. Okay, so here tear level increases like this, and then reach to the plateau here. okay, plateau. And so it means tight junction formation have been completed on the membrane in the microfluidic device. So here the same observation, so microelectric electron microscopy observation also suggests the tight junction like this. So here nucleus, and here is the boundary of the epithelial cells, but no space, and so it suggests the tight junction. And also we checked the immunostaining, and these green lines, the boundary of the epithelial cells, and so these molecules are ZO1. ZO1 is also a molecule related with the tight junctions. And then so completely sealed, and to form that. So it means that no hole. Okay, and also this video shows the objective lens. So here the sample, and then so focusing now here, and then so objective lens being moved from the bottom to the top a little bit, and then focusing point is changing from the bottom to the top. Okay, the focus. So you see, well, yeah. You see, gradually. So first of focus on here, and then gradually you can see. So it means for just so first. So maybe you thought this is a hole, but objective lens move to the top, then focusing point is changing, and then so you can see the cells. Just a moment. It's moving or not? Very slow, or maybe stop here. Yeah. So not hard. So you see the cells here. Okay, gradually. So it means so this video suggests indicates that this area is not whole. It's like this. So this shape or this morphology is very similar to the intestinal villus, like this. And then so I checked another. So I show you another video. Here, you see this villus appeared and disappeared, appear and disappear, like this. Okay. Then anyhow. So after completing the tight junction of the epithelial cell layer, we added the E. coli bacteria, to the upper channel here, blue line. So blue channel. So upper line. And then so both fresh media continue to flow. Then so epithelial cells were able to survive, of course, because here tear, so a little bit of the increase, but so decrease. It means could not survive. So this is a static, so no flow. So no flow. E. coli growing up, and then producing some toxins, LPS or something, and then kill the epithelial cells. And then that's why tear levels goes down. On the other hand, by giving the flow, so tear levels maintain or little bit increase. It means epithelial cell could survive, and the tight junctions were also maintained, at least within 48 hours. And E. coli was not able to invade. Invade means this E. coli, we are afraid of the invade into the lower channel. But you see, of course, upper channel, we could detect E. coli, but on the other hand, lower channel, no detection of E. coli. So on this data also indicate the tight junction had been maintained, and also so by flow. So also maybe you are afraid. So you are afraid of E. coli. E. coli also was disappeared, flow out. But some E. coli stay here and then proliferate a little bit. And then that's why after 48 hours by flow, flow, then so still increasing or stay there. It's very important. And same observation also, you see, this is the epithelial cells, and these are also E. coli here, but no invasion. So I forgot tell you just a moment here. So you see, this face is apical. So apical face of the epithelial cells, they have microvillus here. You see many microvillus here. So it's a typical of epithelial cell surface of the surface. Okay, and then yes, and then so using this device. So I show you another example. So this one is very special. So by collaborating with Dr. Ogawa, so he produced very special E. coli. So normal, so wild E. coli, so healthy E. coli, I mean, has E. coli has no cilia, no cilia. Cilia is like a so not hair, but anyhow, so very fiber, many fibers outside of the cell wall, bacterial wall, and the cilia. So no cilia. So wild type healthy E. coli has no cilia, but toxic E. coli, for example, 0157 is a very popular toxic E. coli. These type of E. coli have cilia to form the biofilm. You know biofilm? You know biofilm. Maybe so. Most of students are working on the bacteriology, so maybe so. And you should know the biofilm. Then so biofilm is a very bad situation, and then but anyhow, but the E. coli, normal E. coli has no cilia, and then so cannot form the biofilm, and then so that's I think one of the reasons E. coli is a very small population in the intestinal microbiota. So E. coli is very famous in the world, especially people using the kanji. So maybe so daicho, dacho, no, I don't know, kid. So it's typical also large intestinal bacteria. So maybe someone most general people believe E. coli is a major population in our intestinal bacteria, but it's not correct. A very less population, and then because E. coli, normal healthy E. coli has no cilia, and then so cannot form the biofilm. That's why. But on the other hand, 0157 E. coli is a toxic virulent E. coli. Virulent E. coli generally form the biofilm because they have a cilia. Then, but Dr. Ogawa, he knocked out RNST2 gene in the wild E. coli. Then, so this E. coli, surprisingly, can produce a cilia. So this delta RNA E. coli has lots of cilia, so ciliated E. coli. So his hypothesis is that this ciliated E. coli would be able to produce the biofilm that stay in the intestine. Then, so he tried like this. So these mice received I forgot the English name. Just a moment. Very simple English term, but I forgot the name. Just a moment. Yeah, basic. Antibiotics. So receive much antibiotics, and then remove all bacteria in the body. And then so after the treatment, then so wild type or ciliated E. coli was administered from the mouse. Then so you see here, so wild type E. coli almost it's very hardly stay in the intestine, but on the other hand, this ciliated E. coli could stay in the intestine here. So this one is in detail one. So you see here many ciliated E. coli exist here. So, okay. So mice given a high doses of antibiotics and to completely eliminate it first, the intestinal bacteria, eliminate the intestinal bacteria, and then each was given a wild type or RNST2 knockout E. coli from the mouse. Then so compared to the establishment and the growth of a wild type E. coli, harness to knockout E. coli can be seen to so establish and grow in the mouse gut. Okay, and so anyhow, but in these animal experiments, the mice have been overloaded with large doses of antibiotics. Can you imagine? So I think, of course, he got the permission from the institutional committee of animal experiments. But anyhow, of course, at that time, he described maybe only one way to investigate this research at that time, at the moment. Then so he got the permission from the committee, in the institutional committee. But anyhow, so very so abundant antibiotics was administered into the animals. For example, also, I don't know, you have a pylori bacteria in the stomach. And then, so if you have a pylori bacteria, of course you have to remove this bacteria by taking the high dose of antibiotics. Fortunately, so I don't have. And then, but my friend, he had, and he received. So he took the high dose of antibiotics every day, or I don't know. But so very much. So he told me. So just I took antibiotics. It's so I eat antibiotics every day. Too much antibiotics. And then so also the same situation. Anyhow, so it's very hard. But now I invented the co-culture model of intestinal epithelial cell and intestinal bacteria. Then so I use this one. So here wild type, and here addition of CD8 the E. coli. First look at the right one. Oh, no movie. So next one, I hope movie. Strange. Just it's a very important slide. So, but movie cannot move. Just a moment. It's a very important. I will check it. Maybe I will show you the another file. Yes. Very strange. Hope this one. Okay, same one. Then so first one. So please look at right one. It's a wild addition of wild type E. coli. You see. Did you find something? No, maybe no. No, of course no. It's okay. On the other hand, ciliated addition of a ciliated E. coli here. So you can find the colony of the ciliated E. coli like this. It's like so this one is a bubble, but anyhow. So it's a biofilm. So ciliated bacteria, ciliated E. coli could form the biofilm and to stay there on the epithelial cell layer. So it's a real alternative of animal experiments. So animal experiments, so we have to add excess antibiotics into the mouse, and maybe so it's a very awful. So mouse, of course, yeah, I can imagine. But using this device, and so we can use this device, and so we can we would know the same result. Okay. Back to the this one. Unfortunately, so yeah. And then next, another example. So inflammatory bowel disease. So inflammatory bowel disease is also very popular in the world. And then so I don't know, maybe we are not suffered from the intestinal bowel disease, because if we have intestinal bowel disease, and so we have to go to the toilet many times, and also sometimes bleeding, and then so it's a very dangerous situation. And anyhow, so so I will read. So a condition where there is chronic inflammation in the gastrointestinal tract, and intestinal epithelial barrier damage causes a prolonged inflammatory response by the immune system from the interaction with uncontrolled entry-exit of content. So this disease completely depends on the immune response. Okay. And however, the mechanism underlying intestinal epithelial damage remains unclear. So, but this disease, anyhow, so is depending on the immune system, but the mechanism still unclear. Okay, and there is model. There is some model. So so there are some papers, and but using these models still have not been elucidated. So mechanism has not the mechanism of the IPD have not been elucidated. So inflammatory bowel disease, we call IBD. Maybe I didn't tell you. So inflammatory bowel disease, so we call IBD. Okay, IBD. So IBD culture models using the cell culture insert like this. So inserts are also used, but it's not possible to co-culture bacteria. So just we can add LPS is a component of E. coli cell wall, that's it, or carrot bacteria or something. But it's not so good for the elucidation of mechanism of IBD. And also this is a simple schematic schema of intestine and intestinal bacteria. Cannot normally enter the inside of the body. It means blood vessels, of course. And sometimes the intestinal epithelium is damaged sometimes. I don't know. So you have I don't know, but maybe you don't have experience to have a piece to have your pieces with blood, maybe, and then but sometimes anyhow receive the damage on the intestine wall, like this. And so some bacteria, of course, after the damage of the wall of the intestine, and so some bacteria enter into the inside of the body by the damage of the intestinal barrier. And so I think this is a trigger for the development of IBD, trigger. Then my hypothesis is that these intestinal bacteria are recognized by the intestinal resident macrophages, which produce some inflammatory cytokines, such as TNF, IL-1 and IL-6 and so on. Then we made the culture model for the IBD, and then this is already invade bacteria invaded into the body. So here. So we added the E. coli from the lower channel, a little bit, small amount of E. coli. OD is a 0.01 study. And also here we cultured macrophages here on the bottom of the device. So macrophage recognizes the E. coli in patient, and then after the phagocytosis, macrophage would be activated to produce TNF alpha or IL-1 and IL-6, such as inflammatory cytokines. Okay. Then this one is also same and explanation, and also using our intestine device. Macrophages attached to the bottom of the lower channel of the device, and also in co-culture, and in the case, macrophage is a row 264 cell line. So it's a mouse macrophage line, but anyhow, so we also use human monocyte THP1 cell line, but now undergoing, undergoing, and so for this tree culture, but this is anyhow. So next time I will show you. And then therefore, after completing completing the tight junction of a C2BBE1 cells and layer, and the macrophage were added the lower channel, and these cells adhere to the bottom of the lower channel. But these macrophages at the moment not active, non-active state. And then E. coli was added. E. coli was added again in the lower channel. Then E. coli was anyhow pulling the trigger. maybe this one, not so. So I will explain this. Maybe just a moment. Change. Sorry. I will show this one. So here, so these are row cells, so macrophages, and these cyan colored one E. coli. You see E. coli. So E. coli divided, so proliferating like this. So you see E. coli. And then so macrophage recognize and phagocytose E. coli. So eat E. coli. And then so activate. So we could observe like this. Yeah. And then so finally we got this result. So this is a normal one. So just a C2BB1 cell culture. The TR levels is like this. And on the other hand, of course, as the E. coli, we can also, so of course, we can co-culture the epithelial cell and the E. coli, it's okay. And on the other hand, so we, of course, we can co-culture C2BB1 cells and macrophage, it's okay. Little bit TR levels decrease because I told you, so this macrophage at that moment non-active, but little bit active. And then so maybe that's why TR level is a little bit going down. But important point this one. So tree culture model. So tree culture over C2BB1 and macrophage and E. coli. So you see the TR levels dramatically decrease. So this means macrophage eats E. coli, and then macrophage is activated and then producing the TNF alpha and so on. And the TNF alpha attack the C2BB1 cells, epithelial cells, and then killed. Then that's why TR levels going down. So it's very simple, and so this one is also. So then we check the, so maybe it's here. So you see co-culture over C2BB1 row cells, and so here you see the plus E. coli, and so TNF alpha, IL-1 beta, IL-6 is no difference, and I can so IL-6 and MIP2 and IL-8. So most of inflammation cytokines are increased by the addition of E. coli. You see. So these inflammation cytokine might induce the cell death of the epithelial cells. So I think this one is very useful for the screening of the anti-IBD drug. And also now a very simple model, but of course we can co-culture the epithelial cell and microbiota, so many intestinal bacteria. So these are consisting of good bacteria and bad bacteria. Then so we can change the population of a microbiota in this system, and then so we can detect we will be able to detect good bacteria population, and then so it's a very simple treatment for IBD. IBD, I think. And it's very available. Okay, and then so maybe so in the yeah, yes. So morning session is finished, and then so just I will explain now, and then maybe I will send some sentences to you, and then so you can send everyone.
Okay.
Okay.
Or can either you tell the students in the class first, and you send the information to me, and I can.
Yeah. So I will send my sentences. So sentence means so information to you.
Okay.
Then so please send
Send to
everyone.
and yeah, confirm
Okay.
it. Yes.
Okay, so let's start because I have only one time for the last lecture. But first, so I will tell you about your assignment. Okay, and just very simple. And so I have already explained, and also I'm going to explain other style of microfluidic device. So if you will use these devices, so you can choose one of them. So intestine device and or hepatic device or anyhow, so you can use one device. Then please describe. So first, I choose this device. Then, so please tell me the application, your application. That's it. So application, but so I use for the diagnosis. That's it. Not like this. So just so I need a little bit explanation. Okay. So maybe one page. So you can use font size maybe 12 or 10.5 font size on the Word, and then one page. So of course, if you can make the some figures, it's okay. So including the figure, one page, and send me your file to my email address. So maybe you have already handout, and so maybe first page. So you can find my email address. Then so as I told you before, after I would receive your email, so I'm going to reply as soon as possible. But if you don't receive my email, so please send me again. Or okay, I am a student at NC. Okay. So let's start. And so this information, maybe I'm going to send Irene about it, and then so maybe she will send you again, maybe. Okay.
And maybe we can discuss about the deadline to.
Deadline. Yeah, yes, it's important. But I don't know the deadline for your grading.
Oh, generally we have to receive the assignments about 7 days after the classes is over. Yeah, so maybe we have to handling the assignment before let me see August 7th.
Oh,
Yeah, and
okay.
the grade we have to
Yeah, okay. So
receive
we will
in-
discuss later, but anyhow, so maybe you have 1 week or something. Yeah, okay? Yeah, because otherwise maybe you are still young, but maybe you will forget maybe. Okay, so let's start again. So in the afternoon, so in this afternoon, so I'm going to talk about co-culture of epithelial cell and anaerobic bacteria. Okay, so E. coli is so aerobic bacteria, so it's okay. But the anaerobic bacteria, they hate oxygen, and then so we have to control the anaerobic and aerobic environment on the device. So anyhow, so it's a very important point, combining aerobic and anaerobic environments. So we have to control. Okay, and then so we invented this device. So you see, this is a very simple schema of aerobic and anaerobic systems. So as I told you before, most of the microfluidic device made from PDMS. So, but the PDMS is a very strong gas permeable, and then so oxygen can pass through the PDMS materials, and then so it's quite difficult to control, to stop the passing, so oxygen passing, and then so we have to control, and then so our device. So these materials are not from the PDMS. Okay, this device not made from the PDMS. Actually, now I can't tell you exact materials because it's a very important point. Then, so, but anyhow, so not PDMS. But on the other hand, so here, this tube. So this tube is also peak tube, and then so not PDMS, but only this tube. You see, this one made from the silicon. Silicon is like a PDMS. Okay, and then so anyhow, so anaerobic environment is made by placing the anaeropack in this container. So this is a container, and so the device is put in the container, and then so here anaeropack. Do you know anaeropack? Anaeropack is a commercial name. Maybe I don't know the Taiwanese in Taiwan same name or not. So this one is just so remove the materials, remove oxygen. So oxygen is absorbed in this material. So anaeropack is a commercial name, but it's maybe a global name. Maybe you can find the anaeropack. Anyhow, so anaeropack remove oxygen. So absorb oxygen. Okay. And then so to make the anaerobic environment, and but anyhow, so here this lower channel medium should contain the oxygen, because epithelial cells demand oxygen for survive to survive because they have to make the energy to survive. Then so they are using the aerobic metabolism. So not yeah. And then also, but on the other hand, here upper channel. Now we are going to culture anaerobic bacteria. So this media should not contain oxygen. But normally, these solutions, so media, contain the resolved oxygen. And then we have to remove. Then here, so this tube is made of silicone and a permeable, gas permeable material. And then here, no oxygen. And then so this oxygen transfer, so move to this air, and then so not the air, so it's no oxygen, only the maybe most of the nitrogen and carbon dioxide, then remove, so move to the outside here, and then so we can make the media without oxygen here, so no oxygen. Then so we can separate anaerobic environment and aerobic environment. Okay, and here of course so epithelial cells and forming the tight junctions, and then so sometimes so you're afraid of oxygen move from bottom to the top, but these maybe condition concentration of oxygen would be resume so consumed in the cell. Then so oxygen cannot go to this anaerobic area. Okay. Yeah. So silicone is gas permeable, and then so oxygen move to the anaerobic area. But this oxygen also absorbed absorbed by anaerobic pack, and then so we can make anaerobic condition here. And also this tube is a peak tube we use, and the peak tube is also a gas impermeable, and this material is also gas impermeable material, and so we can separate between aerobic and anaerobic. And that's why so anyhow so anaerobic bacteria can be cultured with the epithelial cells with we are using this system. And so like this, so anaerobic bacteria here, and so it's a real one. So this is a photo of our total system for co-culture anaerobic bacteria and epithelial cells. Maybe so you have seen the globe box. Maybe so if you operate anaerobic bacteria, maybe so you operate in the globe box. Maybe. So I don't know the other laboratory, but I found a few globe box in Professor Fan's laboratory.
Yeah.
Yeah, and then so, but so this one is very big, and maybe you don't know, but globe box is very expensive. So Professor Fan has a very expensive globe box, bigger one and a smaller one, so maybe three globe boxes at least maybe. I have seen two.
I think two.
Two,
Yeah,
yes.
in the same.
Yeah,
room.
same floor. Yeah, I have seen, and but very expensive, and but we want to sell. We want to sell this system to all scientists interested in co-culture, epithelial cell and anaerobic bacteria, and then but most of laboratories have no grow box. And so we invented. So this one to use this system. So grow box is not necessary. So just you use this one. Then so it's very convenient. And but of course to keep the anaerobic bacteria, each bacterium for the stock, maybe grow box is necessary, sometimes necessary, but anyhow, honestly speaking, my laboratory also has grow box, but anyhow, so using this culture system, so does not demand a grow box, okay? And then so anyhow, so we try to co-culture epithelial cell and anaerobic bacteria here. So this one, so lactobacillus gasseri is one of the representative anaerobic bacteria. Maybe so. Okay. Then so you see, it's a very beautiful result. So in the case of E. coli, so and the tear levels increasing, but here you see so L. gasseri. In the case of L. gasseri, so static, of course. L. gasseri is a good bacterium, but L. gasseri also kill the epithelial cells. Then so under the static condition, so tear levels going down dramatically, and it means epithelial cells die. On the other hand, by addition of the flow, and then so tear levels completely be maintained. Completely was maintained like this. So and also lower channel, so no detection of an L. gasseri. So L. gasseri could not invade from the upper and to lower channel. And so we succeeded to co-culture epithelial cell and L. gasseri. And also we checked bifidolongum. So bifidolongum also famous anaerobic bacterium, and then so we could co-culture epithelial cell and bifidolongum. So results are same. So it's the same. Same as lactobacillus co-culture system, co-culture result, and then so we now fecal bacteria also, and then so it was a little bit difficult to maintain the population of a microbiota on this device, because so maybe human baby gradually taking the several bacteria daily, and then finally the baby succeeds to get the microbiota population. Then so we have to be patient to establish the microbiota population. Maybe needs several days or nowadays for a long time. But anyhow, so but it's possible. It's possible to co-culture epithelial cell and fecal bacteria. It's possible, but the population unfortunately a little bit changed. And then so now we are trying to keep, but maybe we have to add some bacteria first, and then and make the population to make the maintenance of microbiota population maybe. But then so let's move on the next project. So here acetaminophen. So APAP, we call APAP the acetaminophen. So as I told you, maybe yesterday or the day after, the day before yesterday. So acetaminophen is an antipyretic analgesic. So its so effect is so decreasing body temperature and remove the pain. For example, if you have a brain headache and then so sometimes you take this drug. It's a very simple drug and convenient. But you know, acetaminophen, of course, take from the mouth and then so deliver to the intestine. And then we have at the small intestine, acetaminophen is incorporated, and then delivered to the liver first, then circulated. Then so, but acetaminophen mainly receive the modification with, for example, glucuronic acid or sulfuric acid. And then we call this modification is a conjugation. So glucuronate conjugated acetaminophen or sulfate conjugated acetaminophen. So these are become to hydrophilic, and then so they are excreted to urine. So urine at the kidney. Okay. And then so body pharmacokinetics. So in the blood, so acetaminophen blood concentration is gradually decreasing. Then so that's why maybe you take acetaminophen maybe 3 times in a day, okay, morning, lunchtime, and night. So 3 times a day. And then to keep the level, so blood level, so blood level of acetaminophen can almost maintain. And then so, but unfortunately, we are aging. Then so kidney is have been working after the birth. So you have already so your kidney have been working already for more than 20 years, and then so little bit tired, so become tired, and then so aged people. So most of the aged people have a defect of a kidney. Then so this one is not so bad, but looking at the drug, so these conjugated acetaminophen cannot be excreted at the kidney anymore. And then so these these are so circulating, and then back to the liver, and the liver think, oh, why you come back here? liver cell. Why? And then so, but kidney rejected, rejected me, and then so that's why I returned to liver, and the liver said, oh no, okay, so I changed, excrete, so discard you to bile duct. So bile duct connected to the upstream of our small intestine. Okay, it's a very big point. So acetaminophen, and then received conjugation. This conjugated acetaminophen normally excreted into the urine, and then so already so bye bye. It's okay, but return to the liver, and then so change the modification. Molecules are the same, but so these are excreted into the bile duct, and the bile duct connecting to the upstream of the intestine. Okay, so this right, and so then, but so downstream of the small intestine, and also so there are so many intestinal bacteria, and then so these bacteria have beta glucuronidase. So beta glucuronidase digest this conjugation, and then so naked acetaminophen appears again in the small intestine. That's a very big problem, because this acetaminophen, and so naked acetaminophen, can be incorporated at the small intestine again. And then so naked acetaminophen delivered to the liver and then circulated again. So young people, it's okay. So maybe can be maintained like this. But aged people, maybe so not excreted, so return to the naked acetaminophen again, and then so little bit gradual, so increasing. If they take the 3 times after 3 times a day, and then increasing, that's a very big problem. So you are still young, but maybe aging also is starting, and then so. And also in the world, now aged people are increasing, and so looking at aged person people, and then so we have to think about it. Then, but it's quite difficult for checking in animal testing, and then so now we are trying to indicate this phenomenon like this. So anyway, so this graph is so we added the glucuronide conjugated acetaminophen, maybe 100 micromolar in the culture. Then measure acetaminophen. So it's a little bit difficult to directly measure the glucuronide conjugated acetaminophen. Then so we measure the acetaminophen by HPLC. So it's not so difficult. And then of course, starting point, so no acetaminophen now, just conjugated acetaminophen. But 6 hours, so dramatically increase acetaminophen. It means glucuronide conjugated acetaminophen was disrupted to acetaminophen, and then going up like this. So within 24 hours, 30% of glucuronide acetaminophen back to the acetaminophen like this. So it's a very important point, and so now, but of course we want to check the effect on the liver. Then so now we are trying to connect each other, liver chip and gut on the chip like this here. So I explain. So acetaminophen addition maybe I don't know here, maybe here. Then so acetaminophen acetaminophen does not receive the effect by bacterium, and so incorporated anyway. Then this acetaminophen delivered to liver like this, and then liver at the liver acetaminophen is incorporated here like this, and then glucuronide conjugated acetaminophen here, and then in this case sometimes back to him and back to here, but some of them now of course excreted to the bile canalicula. So this is a bile duct, and then so bile duct, and then we go to the intestine again, upstream of the intestine. And then so intestinal bacteria digest this glucuronide conjugated acetaminophen to naked acetaminophen. So naked acetaminophen incorporated again here, and then return to the liver. Return to the liver here. So so I think. And then so we can check the liver injury here, and then so I think it can be alternatives of animal experiment or human test. Okay. Then finally, so but anyhow, so I will show you the how to operate our device. Maybe so after October, I will bring our system here, but anyhow, just it will show you the how procedure on the manipulating our device here. Okay, I hope working. So before starting, so here you can see the device. So you can see the electrode here. Oh yeah, here also. I think it's clear compared to my display. Was small, yeah, okay. Okay. And the hands, not mine, so it's a PhD student's one. And okay. Of course, we have to keep the clean area, but just now in the video, right? Okay. So device and she's checking no bubble because bubble is very very bad. At that time, she used long tube, but now very short. She uses very short tube. You see, this is a micro perissa pump. And here also another perissa pump. No, yeah, this one is a perissa pump. It's not a perissa pump. It's just for the electrode. So left apparatus also necessary. So the total system. Yeah, that's it. So maybe operation is not so difficult, right? And anyhow, so we have to avoid bubble formation, formation, because bubble is very bad because medium, how can I say, so medium channel of course have a height, but if the bubble form, and then so this area very less medium, and it cannot supply the nutrients, and it's a very big problem. And sometimes cells will be killed by bubble. Then so we have to avoid the bubble. It's a very big point, and then so always checking the avoid the bubble coming, and also this operation should be hurry up, because solved air, so oxygen and so on, solved air is of course easily, so this air air is easily resolved in the medium at a lower temperature. And then, so before starting this setting, so we have to warm this medium. And then, so remove the so and then so remove the air. But if the if you take the long time to for the setting, then so medium temperature going down, because warm almost at 37 degree, but of course, for example here maybe 25 or 24 degree, and then so it's easier to be resolved for air. And then so we have to set these materials immediately. It's a very big point. So yesterday we received this question from the other company, and then so my PhD student answered. So you have to finish within 15 minutes. And so she said, so company person took 1 hour. And so she told her so within 15 minutes. It's possible. So but anyhow, so it's very important. And then anyhow, so, and then yesterday, first day, I introduced circadian rhythm. Then so we our system also sometimes have circadian rhythm. Okay, circadian rhythm. And so I have already explained circadian rhythm like this. Like this, and then so microfluidic device has a very big advantage for the stimulation of the drug also. Just a moment. Here, sorry, it's just remove the sealed one. Sealed one is a stimulator drug. So just 20 hours. So maybe stimulation is a very short, 30 minutes. And then so 30 minutes automatically add sealed one. Then change to the fresh medium, other medium at 30 minutes, and then so change automatically. And it's a very convenient. And then so we added this stimulation daily. And then to have a circadian rhythm. It's a very convenient. And anyway, so now we have a heart on a chip, and a liver, and a pancreatic on a chip, and also co-culture of our intestinal epithelium and microbiota on a chip, and also we have also a dialysis device also, and then so now we are trying to connect all together. But can you imagine the problem for this? We have a very big problem now. So, what's this? Problem. What's problem? Of course, there are several problems, but the main problem is media.
Nutrient decrease,
Hmm?
the nutrient decrease along the flow.
No, it's not so. I think at the moment, nutrients decreasing not so big problem because we add very excess nutrients now. It's okay, but for example, liver tissue are cultured in the Williams E base medium. And but intestine device we use DMEM. And heart also DMEM. But nerve chip, no brain here. Very strange. No brain. I forgot to add the brain device. But so nerve tissue are completely different special medium. Then so we have to look for the adequate so common medium. But it's quite difficult now. Then so now yeah. Then so I would like to discuss with Professor So. Here. So he is a specialist for the media, producing the media, and then so maybe so. I don't know the song pronunciation. I don't song so. I don't know. He made he make a venture company. I think a very big company now. And so he's this company is selling the media for the human IPS and so on. And then so he's a specialist for the media. And then so I'd like to ask him. And anyhow, so the problem is media. It's quite difficult. So co-culture model always will face to a media problem. For example, so bacteria, intestinal bacteria also. So each bacteria maybe prefers each media, maybe change. So E. coli maybe you use L. plus, and other lactobacillus maybe is cultured in the other media. And so sometimes yeast extract components, yeast extract necessary, but the yeast extract is very bad for the mammalian cells. Then now we culture the intestinal bacteria without the yeast extract. And then also, so anyhow, so we are always thinking which media or which component is the best or something. It's a very big problem. So anyhow, so almost finished, and then so now this is the chip. Then so I open this chip using this knife and open it, and then inside of the chip, so there are many organs here. So we are now trying, and then also of course this system can replace, replace animal testing and also some of clinical studies also. I hope, and then now we are trying. Anyhow, so still we have several severe problems, but anyhow, microfluidic device device technology has a very big advantage. And also, how can I say? Anyhow, so most of persons who are working on the microfluidic device, these they are always just cultured cells on the microfluidic device. Maybe these experiments just they can use a dish, almost same. But in our case, so we have succeeded to establish the, for example, co-culture model. It's impossible to use a dish or cell culture insert, and it's very important, I think. Okay, anyway, so almost finished, and so as I told you at the beginning of the after session, so please send me a report about for what? So if you use my one of the my microfluidic device, then so you please give me your idea for application as an application. Okay, maybe deadline. Anyway, so please send me anyway. So this weekend or something. Okay, just one week.
Is the reference need to be put in the?
Yeah, it's better. It's better. It's better. So if you want a high grade, so please show me the evidence, some evidences. So using maybe by citation or something. Okay.
Maybe the reference is not included in the one page.
Just mention paper or something. It's okay. And yeah, but anyhow, so just a very simple one. It's okay. Maybe so I don't know maybe everybody understand my talk, and so it's okay. And then so maybe so I grade you not so low. Okay. Yeah. But I don't know the other professors. But yeah. Okay. And so almost finished. So do you have a questions?
I'll ask the online students.
Yeah. Do you have any questions? Of course, not only sending the report, and also if you have some questions, please send me an email. Okay, and also if you want to come to my laboratory, please send me your email. So I'm waiting. Okay. But maybe so I'm going to continue to do this type of work here also, and then so it's okay. But if you are interested in Japanese culture or something, and it's a very good way to come to my laboratory. But if you come to my laboratory, please work harder. Okay.
Sure, and
okay.
online students is there's a question,
Mhm.
but she's,
Just, yes.
yeah, but she's not yet posted, so I'm waiting for her.
Ah, okay.
Yeah.
Will cell will form the villus-like structure in the model, right? I wonder if the number and the density of the villus-like structure will influence the outcome of experiments or not, whether it will influence the outcome.
Maybe
I will ask her again.
beginning of
Sorry,
her talk, maybe I couldn't hear.
sorry, can you say like, yeah, repeat your question again?
Oh, sorry. Maybe, can you hear me?
Okay.
Yeah.
I want to ask a question about the villus-like structure that the epithelial cell form in the model, right? The epithelial cell will form the villus-like structure. I heard that in the morning.
So you are asking about liver microfluidic device, and then but I can't hear you.
I want to ask about the intestine device.
Intestine device, okay, and then.
And the epithelial will form the villus-like structure, right?
Just a moment. I moved my slide.
And there's a maybe microscope image of the villus.
Virus. Yeah. Virus. Oh, yeah. Okay. Virus. Yeah. So your question about virus
My
formation.
question is whether the number and the density of the villus will influence the outcome of the experiment.
Yeah. Okay. It's quite difficult, but anyhow, so to form the tight junction, so epithelial cells should proliferate to reach to the some cell numbers, but I don't know exactly, so I can't answer. But anyhow, and then so after completing of the epithelial cell, so completing of the tight junction, and then so villus structure appears. But I don't know the cell density. So cell density should be high, but normally reach to the plateau to the TR level, and then so proliferation also stop. But so density, I cannot answer. Is it okay?
Okay.
Yeah, okay. did she hear my answer, my voice or something?
Because the site is still still working.
Okay, other question? Okay. So, thank you very much for your attention, and so I'm waiting for your reports, and also if you have other questions, please send me your email, right? Okay. And also, if you can, so please please write your opinion to my lecture also. Okay. Yeah. It's optional. Okay. Thank you very much.